Welcome back to the pharmaphorum podcast. In this episode, I speak with Rahul Kakkar, CEO of Tome, a gene editing company that launched last December. From a brief history of biopharmaceutical innovation to an overview of the moral implications of gene editing and how applicable industry's frameworks are for such, Kakkar provides insights into recent developments in genomic medicine, including Tome's programmable genomic integration technology, and why pricing and reimbursement of medicines must be value-based, moving with the times and re-envisaging drugs with the potential to provide decades of efficacy, a sea change in reimbursement applicable to the sea change in science. It's a wide-ranging conversation, but hopefully one of value for those seeking nuggets of insight in this field. And, as ever, thank you for listening. This is web editor Nicole Raleigh, and today I have with me Rahul Kakkar, CEO of Tome, a new gene editing company that launched in December 2023 with $213 million in Series A and Series B funding to advance its programmable genomic integration, or PGI, platform. PGI enables the insertion of any DNA sequence of any size into any programmed genomic location, and Rahul himself has termed the technology revolutionary in its ability to, and I quote, reprogram the human genome with an elegance and efficiency previously unimaginable. Indeed, PGI combines the site specificity of CRISPR-Cas9 with enzymes capable of inserting or rewriting sequences of DNA, including entire genes, without the need for double-strand DNA breaks. But I'll let Rahul himself go into the finer details of this as we discuss the future of gene therapy in our conversation today. So to begin with, Rahul, perhaps you could tell listeners a little bit more about yourself and the passions that have driven you to now and the work you are doing with Tome.
Thank you so much, Nicole, and I appreciate the opportunity to be speaking with you and your listeners on your podcast. Yes, Tome, as you described, is really a technology company that is based on a revolutionary way of editing DNA. I came here just before the first round of funding closed back in 2021. This is my third company within the industry, and it was really a recognition of the potential of the technology out of the founders lab at MIT that drew me to Tome. It's very rare in our careers that we get a chance to be involved in a company or a technology or a methodology that can really redefine the way we develop medicines for patients. And that's what Tome represented to me at the time and continues to this day.
So defining the way that medicines are created for patients.
That's right. If we take a big step back and think about our industry, really since the 19th century, I would say that there have been three horizons. And to me, Tome sits at the forefront of the fourth. And what I mean by horizons is the first was really in the late 1800s and into the early 1900s, with the rise of synthetic chemistry, no longer needing to use natural elements to create medicines, but being able to synthetically create them in the lab, what we call today small molecules. The next horizon was the rise of our ability to clone DNA and create proteins from them, which gave the rise of the biologics industry, injectable therapeutics that we use today, whether they are antibody-based drugs, insulin, or even some of the peptide-based drugs that we use for weight loss today. The third horizon really is very recent. And really came to the fore during the COVID era with the rise of nucleotide-based therapeutics, either for controlling pathways, companies like Alnylam, or for vaccines like Moderna and BioNTech. But we haven't yet, I don't think, seen the rise of a company and technology that can truly define genomic medicines the way companies that we think of today have defined synthetic small molecules, large molecules, and RNA therapeutics and vaccines to date. And I think that's largely been because our ability to edit the DNA has been very, very limited. Has all the promise in the world, even with the discovery and the Nobel Prize winning science around CRISPR-Cas9, it still is very limited in its native form in terms of our ability to reprogram DNA at our whim. And when I saw the technology coming out of MIT, which combines, as you mentioned accurately, the power of CRISPR-Cas9 with other enzymes to augment its capability, at least in my imagination, I saw the rise of a technology that could truly define genomic medicines or medicines that are derived from our ability to manipulate DNA.
Okay, thank you for that. Let's call it a pocket history, if you will. But what about perhaps the ethical issues that history into the genomic medicine side of things has faced along the way? Can you comment on that at all?
There are very serious ethical implications, moral implications for us as an industry and us as a species as we think about beginning to manipulate the code of life, for lack of a better word. I think that is why it's appropriate for these technology to be developed within a very regulated framework like the biopharma industry, where uses for improvement of health and curing of disease is the stated goal of both regulatory bodies as well as the investment community, and not for potentially untoward uses that one could potentially imagine.
Yes, absolutely. Okay, so if we focus on Tome itself, specifically its most advanced PGI technology, the integrase-mediated PGI, or iPGI, which is based on the PASTE, or if you want to spell it out, PASTE technology, first discovered by co-founders Omar Abdouier and Jonathan Gutenberg while investigators at MIT. Can you, Rahul, tell us more about how that works, why it is, as it has been called, a game changer, and its intentional use for enabling future complex cell engineering, including for monogenic liver diseases and autoimmune diseases?
By all means. In order to understand why PGI is a concept and then integrase-mediated PGI, and we can also talk about ligase-mediated PGI are such a step forward, it is important to look back and say, how have we approached creating medicines through our ability to manipulate DNA to date, and why has that been so limited? And by and large, when we want to create a medicine that changes the way the cell behaves at the genetic level, we have had to do one of a few different things. Either place a gene into the cell that never integrates into the genome. It functions, for lack of a better term, like a computer subroutine, functioning on its own within the cell, but not part of the cell software itself. This is traditional gene therapy today, and it has certain liabilities. The gene is not regulated by the cell at all, and if the cell divides, that gene is lost over time because it doesn't replicate with the cell itself. There's a few other ways that companies and academic researchers have attempted to move the field of genomic medicines forward, trying to integrate those genes into the genome. But to date, it's always been either randomly integrating or integrating into certain what have been called safe harbors within the DNA, or areas where we don't believe, at least, we're going to be breaking the DNA when we put the gene into it. Obviously not elegant, right? Elegantly, you would want to actually put a gene in a location where the scientist or the drug developer designs it to go and not have it randomly integrate into the genome somewhere. With the rise of CRISPR technologies and even iterations of CRISPR technologies like base editing and prime editing, we've moved to being able to very site-specifically edit the DNA, but only with very small edits, single base pairs, single letters, a few letters at a time. Again, it sort of it solves the integration problem, but it now creates a size problem. Most genes are not a letter or two big. They are thousands and thousands of base pairs. So the ideal technology would overcome the limitations of all of these prior technologies. In other words, integrative, site-specific, without a size limitation. And I will only add that it also has to operate at an efficiency that you can edit a significant amount of cells in an organ to actually cure that organ of its dysfunction. And we haven't had a single technology to do all of these things. Integrative, site-specific, large size format with high efficiency. Each technology to date has been missing one ingredient, and PGI, particularly integrase-mediated PGI, overcomes all of these limitations in one fell swoop. It allows us to insert DNA into the genome, thousands, if not tens of thousands of base pairs large, with complete control over the insert in terms of where it goes and how it goes in terms of its direction and orientation, with very high efficiency. And so that's why when I read the original paper from Omar and Jonathan's lab at MIT, the PASTE paper, which we've significantly re-engineered for industrial purposes, which is why we now call it integrase mediated PGI. That's where I said, this is the technology by which we can actually create the mature form of genomic medicines.
I like that expression, the mature form of genomic medicines. So basically, from my perspective, it's the wow factor for this area of science. So going with that sort of propelling wave of positivity, as everyone knows, it was recently Rare Disease Day, and cell and gene therapy certainly hold promise for developing treatment for rare diseases. But there ever arises the problems of cost and access. On the topic of value, though, news broke a little while ago about Ginkgo Bioworks having been on the M&A path, and there was even earlier news this year that Tome itself had acquired Replace Therapeutics, which develops novel small DNA sequences complementing Tome's focus on large DNA integration technology. So the appetite is there, and I think it's fair to say that the promise of these therapies is there for such to be so. But Rahul, can you tell us any more? I mean, how much does all this truly expand the potential types of diseases that can hopefully be cured?
It's a phenomenal question. There's a few very important points in the question that you asked, Nicole. One, and I made this comment the other day when I was speaking to someone. I don't come from a scientific background in cell and gene therapy or DNA editing. I come from a clinical background. I still see patients here in Boston, in one of our local hospitals. And when I look at novel technologies coming from academia or being developed in early stage biotech in our industry, my first question is, why would a patient care? If you're not delivering a medicine that is differentiated, specifically offering better efficacy or better safety, or ideally both, than the way we treat a certain disease today, then it doesn't matter to me how incredible the academic technology is. It is fundamentally academic. And I look at many companies in the genomic editing space all going after the same diseases with the same potential clinical profile. And I ask myself, are we truly spending the capital in a very capital intensive environment wisely? And so what we did when we first came to Tome, myself and the chief science officer in particular, now joined by, of course, many others, was look without rose-colored glasses at where could PGI be deployed in a manner that offered patients something truly novel, either a disease that isn't well served now, or if it's a disease that's being served by other molecules and modalities, a true step change in potential efficacy. You use the word cure, and it's not a word that I use lightly. When I started in this industry back in 2011, we never used the word cure unless you were developing an antibiotic. But today, we can begin talking about curing rare disease and even potentially curing, some common diseases through the use of cell therapy. We're unique in the genomic space as a company in that we are developing both gene therapies, what we call integrative gene therapies, and truly novel cell therapies. And that's enabled by our technology. And the diseases we're going after are unique in that they are not diseases commonly pursued by other genomic editing companies. And to me, that is the testament to the potential of the technology. Because if we couldn't make that statement, if we couldn't say that the diseases in our pipeline we're addressing with clear and obvious differentiation for patients in terms of efficacy or safety, then you and I would not be speaking today.
Again, nicely put. I mean, that gets to the point. Thank you for that, Rahul. So, setting aside this fragile word of cure, finally, at what juncture, in your opinion, would or will all this ever-advancing technology be enough to, let's say, crack the code in these endeavors? Will that day come?
So it's already started to happen with the first CRISPR-approved therapy offering cure to the sickest of the sick patients with sickle cell disease. I would say that we're just starting to see the tip of the iceberg of the potential of these medicines. But like any first technology, any Gen 1, and that could be in biopharma, that could be in tech, that could be in AI, that could be anywhere. The first generation is generally awe-inspiring, but far from what we'd like to see. And as you see next generation companies and next generation technologies move forward, what I hope everyone's endeavor is, and certainly our endeavor here at Tome, is to design versions of these genomically derived medicines that are far easier for patients to take, that more patients can access, that offer true value to patients for the cost of the drugs. You had mentioned access and cost, and we can certainly talk about that. But at the end of the day, if we are not offering patients better life, allowing them to be more productive members of society, then we as an industry cannot morally or justifiably demand some of the kinds of costs that are being put on these medicines.
No, this is very true. So do you want to perhaps go down that conversation on cost and access when it comes to these therapies? Is there remit within this conversation today? I mean, it's a big question. It's a big topic.
Yeah, no, I'm happy to discuss it. And this is it's a topic I've discussed with others on podcasts, et cetera. And I have a real internal conflict around this because when I go into the hospital to treat patients on the cardiovascular service, where I attend a few weeks out of the year, it's almost every patient discharged from the hospital that we're wrestling with whether that individual can afford their medicines or not. You know, obviously I practice in the United States where healthcare costs are exorbitant, and there are plenty of stories of patients and their families struggling to afford their medicines because we don't have a single payer system here in the US. And I struggle with that with every patient discharge because invariably I'm having to scramble to switch medicines at the very last minute because the patient's insurance company doesn't cover a certain medicine. And on the other hand, then I come back to Tome and we're developing some of the most complex and from a manufacturing perspective, expensive medicines ever envisioned by our industry. So I do have this conflict and this constant thought process around how are these worlds going to collide at some point? And although I don't have all the answers, what I will say is this. Pricing and reimbursement of medicines, I believe, must be value-based. There are independent bodies, both in Europe and the US, as well as other major territories, which do those sorts of analyses that are not colored by conflicts of interest. And I think it's important for our industry to work closely with such bodies to make sure that we are, from a quantifiable standpoint, only asking to be reimbursed for that value that we give back to society itself. And to do more so, I think, is not morally within our charge. Now, ultimately, going back to the word cure, there's a second barrier, which is, at least in the US and Europe and other more developed healthcare systems, the reimbursement of medicine is based on how medicines are used today, which is on a chronic basis to control chronic disease. Our healthcare systems, by and large, have not envisioned how to reimburse a drug that's given once to provide 5, 10, or 30 years worth of efficacy. And so it's not, to me, just a need to be true to that first principle of value, but it's also a sea change in the environment of reimbursement that has to be complicated if we're truly going to bring these sorts of efficacious therapies to patients.
Yes, a sea change in reimbursement that's applicable to this sea change in science. Thank you, Rahul. It's been an absolute pleasure.
Pleasure's been mine.
And so that concludes another episode of the pharmaphorum podcast. You can find out more information about this episode, including a download link and information about previous installments of the series at pharmaphorum.com/podcasts. The pharmaphorum podcast is also available on iTunes, Spotify, Acast, Stitcher, and Podbean, where you can find and subscribe by searching for pharmaphorum. Of course, don't forget to visit our website itself, where you can sign up for daily news and analysis bulletins, and follow us on Twitter, or X nowadays, at pharmaphorum. That's all for now. Thank you for listening.